my name is Jenni Kao. I'm an undergraduate at UC Riverside, and my project is predicting future Arctostaphylos species distributions using machine learning. The co-authors for this project are Huang, Glen Morrison, Tito Abbo, and Litt. Climate change is an important issue because it is predicted to result in coastal erosion, increased fires due to rising temperatures, and habitat destruction. This is especially important in California because the state has great biodiversity and is already characterized by a dry, fire-mediated climate. The California forested province has chaparral regions and a Mediterranean climate, which consists of hot, dry summers and cold, wet winters. On the right is a photo of the California chaparral biome. For this project, we are looking at the predicted effects of climate change on the distribution of Manzanita species. Part of the Ericaceae family, Arctostaphylos has approximately 60 species, and 49 are considered narrow-endemic, meaning they're restricted to small areas characterized by specific soils and other environmental conditions. As a result, around 44 species are considered threatened or endangered. Most species are found in the chaparral biome, but some also occupy different habitats. On the right is a photo of a manzanita tree. Manzanitas are woody shrubs or trees distinguished by their red bark, curving branches, and bright green leaves. The flowers are urn-shaped and are either pink, white, or a combination of both. They produce small drupe-like fruits that are said to taste like apples, hence the name manzanita, which translates to little apple. The objectives of this project are to predict future Arctostaphylos species distributions using machine learning so that these predictions can facilitate conservation efforts. I chose 3 narrow-endemic species from California: Arctostaphylos cruzensis, otaiensis, and perissima. The reason why I chose these species is because of their different habitats. Arctostaphylos cruzensis is found in the San Luis Obispo and Santa Cruz area. This habitat is considered a maritime chaparral or coastal prairie and has occasional short-term coastal marine fog in addition to the normal Mediterranean climate. Arctostaphylos perissima is found in the Santa Maria and Long Park area. This environment is a combination of coastal sage scrub, chaparral, and oak woodland. The climate is slightly different from the normal Mediterranean climate since it has short, warm summers. Arctostaphylos otaiensis tend to grow in southern San Diego, specifically the San Ysidro Mountains. Characterized by coastal sage, chaparral, and footland range, this habitat has mild, wet winters and hot, dry summers with occasional fog. This plot shows the general distribution of each species. The primary method of this project is to use species distribution modeling to predict the occurrence of manzanita species. This form of machine learning investigates the relationship between species presence and various environmental conditions. Using algorithms and models, we can use species occurrence and environmental data to predict future environmental suitability based on predictive models of climate change. This figure comes from conservationbytes.com and illustrates the process of species distribution modeling. Using environmental data from the present, they are able to predict the future environment to see how niche distribution might change. The occurrence data used in this project come from the Consortium of California Herbaria database, which is a collaborative online database that consists of specimens from various California herbaria. I use specimen data for each species from April 1970 to April 2022. All climate data came from WorldClim, a global weather and climate database. For historical data, I use climate data from 1970 to 2000. WorldClim also contains predictive future climate data, so I chose to look at the years of 2021 to 2040. I use the Earth System model from the Euro-Mediterranean Center on Climate Change. Within these models are shared socioeconomic pathways, or SSPs, which act as inputs for these climate models based on predictions of the impact of climate change on factors such as population, economic growth, education, urbanization, and the rate of technological development. Each SSP is based on a different prediction of temperature change. Each dataset contains 19 bioclimatic variables such as annual mean temperature of the driest quarter, and annual precipitation. The spatial resolution for both datasets was 30 seconds. To investigate how climatic conditions affect manzanita species, a jackknife test was run to calculate the importance of each variable in determining a suitable habitat. In each run, one variable is excluded and the remaining are used in the model. The teal or longer bar represents the probability of a suitable environment without that variable. The blue or shorter bar is the probability with only the tested variable. Here I am showing the results for one species, Arctostaphylos cruzensis, but the results were similar for other species. As you can see, you can leave out any variable and the remaining data will still accurately predict environmental suitability. No one variable is critical. However, some variables contribute very little, such as BIO2 or mean diurnal range for Crescensis. We can identify some variables that make a more considerable contribution, like BIO1 or annual mean temperature and BIO11, mean temperature of the coldest quarter. When we look at the other jackknife results for the other species, we see that for Arctostaphylos parissima, variables BIO1 or mean temperature of the coldest quarter and BIO19 or precipitation of the coldest quarter contribute a significant amount, whereas variable BIO14, precipitation of the driest month, contributes very little. For Otaiensis, variable BIO4 or temperature seasonality and BIO14, precipitation of the driest month, play somewhat of a role in habitat suitability, while variable BIO16, precipitation of the wettest quarter, does not. To predict Manzanita distribution, I use the program MAXENT, which is short for maximum entropy. This program calibrates to find the most spread distribution, meaning it finds the maximum probability of environmental suitability while considering ecological constraints. MAXENT requires species occurrence and environmental data to investigate where the environment is suitable for the species after training on the current species distribution data. Warmer colors show a better environment for the species, and the white boxes are the presence data that the model trained on. The plot on the far left is the suitable habitat for the historical and current climate data of Arctostaphylos otaiensis. The other plots are the predicted suitable habitats based on the 3 different SSP scenarios. In all 3 scenarios, the Arctostaphylos otaiensis habitat has a high probability of expanding to the north and east. Arctostaphylos crescensis habitats are estimated to expand the most out of all species in every SSP scenario. The most suitable habitat is still predicted to remain on the coastal regions, expanding northwards towards Washington state and southwards into Mexico. Additional suitable habitat is predicted to develop in the east as well. Arctostaphylos parissima habitats are not predicted to expand as much as the other species, although they are expected to expand up and down along the coast. The modeling also predicts highly suitable habitat inland along the foothills of the Sierra Nevada. In conclusion, the habitats for these 3 Arctostaphylos species are estimated to expand based on predicted climate data, but this does not mean they will end up in these regions. Based on these predictions, it does not appear that climate change itself is a threat to these species. However, other threats still remain from fires and human activity. It is possible that if northern Manzanita species become extinct, southern species are able to fill these ecological niches. Thank you.